First up, Xiaoqian Chai, it's over to you.
I'm very happy to be here today to have the opportunity to share with you guys my research. Title of my talk today is the hippocampus, default network, and memory development. To me, the process of child development is really fascinating because compared to other animals, human babies are born exceptionally underdeveloped. For example, there are many animals like horses and giraffes. The baby animal can walk basically within minutes of birth. Compare that to a human infant. Here's a picture of my son. I think he was already like 13 months old, had no motivation of trying to learn how to walk. He was just crawling around happily because he couldn't depend on me. I guess the trade-off of such a immature baby with an immature brain is that we humans have then developed this amazing capacity to learn many skills that animals cannot achieve. So it seems like the humans have evolved to allocate more time for brain development. If you look at the human brain, it takes something like 2 decades for it to be mature to more like an adult state. So during this like 2 decades, we are able to acquire these really amazing skills. So my research overarching goal is basically to understand how brain maturation relates to cognitive development. The main method I'm using is fMRI. So here I wanted to just thank the McConnell Brain Imaging Center for their amazing support. So they let us really decorate this mock scanner that used to be a 1.5T MRI. So we made it child friendly, and we've recently started collecting data, including kids as young as 5 years old. And we're also testing some children with autism. So for today, I was hoping to share with you some of the results and ongoing research on how functional brain maturation supports memory development. And we are focused on the hippocampus and also how it interacts with cortical networks during these memory processes. And as I mentioned, we just started a project looking at memory and social cognitive development in autism. I'm hoping to get feedbacks from you guys if anyone has any comments, ideas, suggestions, or ideas for collaborations. Let us know, because some of this work we're still trying to write up for publication, and then the autism project is just started. So when I talk about memory development, I really mean episodic memory, which is a memory of past events or experiences. So if you think about, a specific event you had, for example, you had a dinner party someday, it's actually an integrated experience of many different pieces of information. So you can have memory about, what drinks were served, what food you ate, and also more of these a situation or context of the event. For example, like what was the occasion? Was it a lab gathering, a birthday celebration? In terms of development, there is pretty rich behavioral literature in children, which basically supports the idea that our memory for specific items or familiarity develops relatively early on. So by age 5 or 6, it's pretty mature. But it's really the ability to recall these more complex or contextual detail information. For example, what was the occasion, who was there, and when. So these memory details, children actually don't encode them very well. So you would see an increase in ability to recollect these episodic details all the way till about like mid-20s. And then sadly, after 30, it goes all downhill. So, the episodic memory is supported by the hippocampus and several, large-scale networks. So exactly, how these different pieces of information are encoded and supported by where in the brain. So that's still somewhat hotly debated. But so one large-scale network that's really relevant for episodic memory is the default network, and I'm sure most people here are very familiar with this network. But basically, you would see default network activations for things like autobiographical memory, episodic memory retrieval, and also for social cognitive processes like theory of mind tasks. You will see overlapping regions in the default network. Here on the bottom panel, I'm showing an image of this notion of task positive and negative networks in the brain. So basically, what you see in red, this is the default network, which people also call it a task negative network. And this is because when we are doing a task that's demanding our external attention, usually you would see this network deactivated or suppressed. So therefore the term negative. And then the task positive regions are the ones shown in here in blue. So these are frontal parietal regions, dorsal attention regions, or basically regions that are supporting cognitive control or attention. So there seems to be some sort of competitive relationship between the task positive and task negative networks in the brain. When one is activated, the other one gets suppressed, and vice versa. And here in this picture is during the resting state, you would see these networks anti-correlated, even though the person is not really actively engaged in any task. So this anti-correlated relationship is really interesting. And we were wondering, when does this develop in children? So in the early study, we showed that they actually develop really late. So here is an example. If you take the medial prefrontal region in the default network, and then you look at the resting state connectivity with the rest of the brain, you would see that, for example, here in the dorsal lateral prefrontal cortex, the connectivity between these regions are actually positive in children, and it just slowly gets developed into anti-correlated relationship in adults. So in children, as if the brain is not really separating these competitive networks yet, and they're kind of mushed together as one network. So what does this mean behaviorally? How does it relate to behavior? So in a later study, we were able to show that kids who have a more immature pattern, which is like a more positive relationship between these regions, those are the ones who would later develop a worsening of attention problems. So this is, again, kind of evidence of this competitive relationship between the default network and the attention network in the brain, and seems to be sort of at least driving some of the development of cognition in behavior in children. So how does this relate to episodic memory? So we did basically 2 experiments to look at this question. So in the first one was focused on the encoding process of episodic memory, because if you look at the episodic process, first you have to encode the memory, and then the retrieval is a somewhat different process. So we looked at these separately. So in this experiment, children and young adults were scanned when they were looking at these pictures, natural scenes. And then after they finished the scanning, they did this recognition test. Basically, each picture was tagged as a remembered trial, or a familiar, or a forgotten trial. And then we go back to the brain to basically sort the time points where they remembered something versus when they forgot something. When you contrast the two, you get the brain activations that are relevant for or supporting successful memory encoding. And this is what we call a subsequent memory paradigm. So in terms of the default network, we did see this expected pattern of deactivation for successful memory encoding in adults. But this again seems to be a pattern that slowly develops from children to adults. The adolescents are somewhat in between. So for example, in the PCC region of the default network, you would see that for a trial to be remembered, it has this region, it gets deactivated more than a trial that was forgotten. So if you look at children, it doesn't seem, this region doesn't seem to differentiate a remembered trial versus a forgotten trial. So it's not really supporting a memory encoding yet in children. And then we also looked at the hippocampus during this task, because hippocampus is sometimes lumped together with the rest of the default network. And here that you see that children actually do use the hippocampus to support their memory encoding. And if you look at the difference between the remembered and forgotten trials, the difference doesn't seem to grow with age. So it seems to be pretty stable from children to adults. For memory retrieval, the story is, I guess, a little bit different, because we know that default network is supposed to support this internal oriented process. Memory retrieval is actually one such process. You are actually focusing on your internally generated thoughts. So in this experiment, we scanned both during the recall. So this is a acute recall paradigm. So participants were shown a pair of unrelated words. And then after that, they were given the first word in the pair, and they were asked to verbally recall the second word in the pair. So during this recall process, we looked at, again, the brain activations associated with successful memory. Here, the pattern is actually sort of flipped in terms of what's higher in a successful memory versus a forgotten memory. So again, I'm looking at the contrast of hit versus miss. And in both children and adults, you see this a lot of activations in the default network, in the medial prefrontal cortex, the PCC, and also these lateral parietal regions. When you contrast the two, so okay, this pattern is again, as I mentioned, what you expect because of the nature of the retrieval process, and that's one of the, I guess, the functions of the default network. So here it's an activation, not deactivation. But if you contrast the two groups directly, it does seem like adult group would have more of these DMN activations for successful retrieval compared to children. So here, if I do the contrast of the two groups, I would see two regions, one in the PCC, the other one in the angular gyrus, both showing more activations for adults compared to children. And if you look at the activations for hits and miss separately, so these bars are actually showing opposite pattern as the from the one from the encoding, where the successful memory here showing darker color. So here it's more deactivated for the successful memory, but here for the successful memory in adults, you actually don't deactivate much. And it's the forgotten memory, you see the more of the deactivation. So the pattern is completely flipped. And in the hippocampus, here the story is similar to encoding, but slightly different. But it seems like children here are using slightly less than adults. But again, this is a positive activation. These two experiments together, I think it's interesting that to show this changing dynamics in the DMN in terms of, during the encoding process and the retrieval process, the DMN seems to be doing the opposite activation versus deactivation. And in children, they don't seem to be engaging the default mode network as much as adults for their episodic memory encoding and retrieval. I don't have a picture here, but these activations, some of these regions are actually supporting how accurate a person would be in their memory ability. The next question we wanted to ask is to sort of look at this like self-related memory encoding. So this is for two reasons. And the first reason is that the default network is linked with these self-referential processes. And episodic memory, as we know, is very intimately related to the concept of self. And we know the concept of self is actually something that develops pretty early on in childhood. So we wondered, if we ask someone to encode something using a self-referential strategy, would we see the same sort of pattern of activation, deactivations, and how does that differ between children and adults? So in this study, we had two conditions during the memory encoding. So they were either asked to think about, if they like this object or this toy. In some other trials, they look at the objects and we ask them, is this a living thing or not a living thing? So they're either doing a self-referential judgment or a semantic judgment during the memory encoding. We showed that, if you encode some information when you're thinking about yourself, you actually are more successful in forming that piece of memory. And this is actually a well-known phenomenon called the self-referential effect, the SRE. So in all the groups, they seem to benefit from this self-referential encoding. And we see that there's actually a very steep increase in the self-referential facilitation for memory in children younger than 11. We see this very strong effect with age. We actually see the age effect across the whole sample, but in the youngest group, it's more dramatic. So there's a big increase. And it's interesting that the self-referential encoding benefit extends to memory for the sort of the background, not background, so the encoding context. So in our task, they were also tested on their memory of the background of the picture they saw, and also the which question they were thinking. Is it the self, I like it, don't like it, or was it the living, not living? So you get boosting memory for the encoding context. So in the brain, it's really it's interesting to me that again, we contrast the remember versus forgotten or hit versus miss. You would see that in the self-referential condition, you some activation in the medial prefrontal region, and not so much. You don't see that in the semantic encoding condition. And in the semantic encoding looks very much like what we saw before, which is a lot of deactivation in the default network, in the midline, also in the lateral parietal regions. In the self-referential condition, not so much. So this is, I think, consistent with what we know about the role of the default network in terms of like internal oriented attention and like self-referential processes. So then the question is, what happens with children? So for this project, we have a continuous age range from all the way from I think 7 to 20, somewhere like 30, 25. If you look at the subsequent memory activation and compare what's greater in the self condition versus semantic condition, and for all participants, you basically see the default network and also the hippocampus, which means somehow the hippocampus is more involved in the self-referential encoding as well. And there's an interesting age-related change in the dorsal medial frontal cortex. This is a region that has been previously linked with self-referential process. And as you can see from children to adults, this dorsal medial prefrontal cortex activation increases. And this is showing that perhaps children, they're not really utilizing the self-referential region to support their memory as much as adults, because this is an activation for the subsequent memory. So it's hits greater than miss. And then we also looked at the functional connectivity of between the hippocampus and the rest of the brain. And only one cluster came out significant in terms of correlation with age. And then it seems like between the hippocampus and the medial prefrontal cortex, there's more connectivity going from children to adults. This is, again, during more for the self greater than semantic encoding. We are just starting this project to look at self-referential memory in autism. We are very excited about this because there's some research showing that suggesting the self-referential process may not be, it may be abnormal in people with autism. And we wanted to know like how this affects memory. And this is actually one of the theories on autism. It's possible the deficits in self-referential process or self-referential memory could contribute to the other like social cognitive deficits in autism. So we, for this project, the task is a little bit different. So instead of comparing self versus the semantic condition, we had a self versus other. So they are encoding these words on the screen, and then they can either make a judgment about whether the word describes themselves or whether it describes the other, which is the minion. We just have some very preliminary results to show today. So when you look at, again, the difference between the self versus the other condition for the control group, it does seem like they are benefiting the self encoding. So if they answer the question thinking about themselves, they're more likely to remember the word. In the ASD group, it's a very small sample, so I can't really draw any conclusion, but so far it looks like they're having less of this facilitation. And these are, I guess, right, so these are children in both groups matched with the age. And we haven't really analyzed the brain data for this task yet, but we have another task in this study. So as I mentioned, the theory of mind network seems to be overlapping with the episodic memory network. And there's some work from Randy Buckner's lab showing that in adults, these two networks perhaps are more separate. They have some overlap, but they should distinct separate networks. But in children, we don't know the extent of the overlap of these two networks. And in autism, we don't know what's happening either. So that's why we added this task. It's a movie localizer developed by Rebecca Sachs's lab at MIT, but it's a very short film, 5 minutes, in which we can use this to identify brain activations related to mental states or theory of mind or pain-related scenes. So my student, my postdoc, Zeus, just analyzed some pilot data from this task. And so far, I think the results look very promising. In neurotypical controls, we're seeing some strong activations, actually in both groups, strong activations in the PCC and some parietal regions, maybe TPJ, that was expected for the theory of mind. In the autism group, it's very small. Sorry, it's a typo here. The autism group is, I think, only 8 subjects or something. So we don't know what's happening yet, but it does look like maybe it's a bit less activation compared to the controls. Tell you about, I guess, 2 studies using existing data. So this is a collaboration with Mallar Chakravarti. So my undergrad student, Shirley, did this work with the HCP-D data, which has something like 545 subjects from 8 to 22 years old. So we used Mallar's maggot brain segmentation for hippocampus, which is supposed to be a pretty advanced algorithm to give us volume measures of these different subfields in the hippocampus. And so far, the results seem to suggest there is a pretty robust increase in volume in many of these subfields, namely the CA1 and the dentate gyrus subregion of the hippocampus. And the boys seem to have a bigger volume with a steeper increase with age than girls. My student Pascal did this study using the Child Mind Institute HBN data set to investigate hippocampal connectivity in ASD. Here we divided the hippocampus into anterior and posterior hippocampus. So this is a mask given to us by Signe Sheldon's group in the psychology department. But we found a really interesting pattern in the medial prefrontal cortex of the default network. So in both the anterior and posterior hippocampus, the autism group seems to show this less deactivation, so less connectivity with the hippocampus compared to controls. And we also found some developmental differences between ASD and controls. So the connectivity between the posterior hippocampus and this precuneus region showed a decrease with age for the ASD group, but in neurotypical controls, it remained stable. We think this is really interesting because the precuneus is this so-called postmedial network, which is supporting episodic memory. And with the posterior hippocampus, we are, I guess, not expecting a decrease with age, which is what we see in the typical controls, but with ASD, seems to go down with age. And unfortunately, the HBN data set doesn't have episodic memory scores. We can relate this to, don't know for sure how this contributes to the memory differences. I think that's all I'm going to show today. And so I'd like to thank my previous mentors, collaborators, and students and funding agencies. And thank you for listening.